बताऊंगा, लेकिन वो जो किचन का जो पूरा
Hello, people. Good morning. Can you hear me?
Hi, Amit. Good morning.
Morning.
Good.
Good
So
morning, Amit.
good morning. So we will wait till 11:35 for others to join. In case if everybody has joined before that, then we can start. Otherwise, we will start at 11:35. Okay.
Okay, Amit.
No, Niranjan, the session has not started yet. We are waiting for the people. Now it's 11:35, so we will start anyways. Those who are left, they can join. So first, Abhi, are you there in call? Are you able to join? Abhishek, can you guys hear me?
Yes, Amit.
Yes,
Yeah.
Amit.
So Abhishek, are you there in call? He had some issues with his number and all those.
Sir, Ram 25.
Abhishek, I don't think that he has joined yet. Who is this Zoom user?
Hi, this is Gaurav.
Gaurav, okay. But Abhishek was
सर
there who was
डोर रुका दे बेटा डोर रुका दे
okay. We'll see as to when he can join. Okay, guys. So first of all, last week we didn't have the session, so sorry for that as I was traveling at that time. Before proceeding, were you people able to do the homework or the assignment that was given, any challenges that you face or any questions if you have just 4 to 5 minutes to ask those, because today's session is very important and it will take some time. So I need to give today's whole time to that session. So if any questions, any challenges from the previous session, if you have, then do let me know. Anyone? Abhishek, I can see now.
Hello.
So I believe no questions from anybody. So let's start with today's session. So as I mentioned that today's session is regarding the risk. So why am I saying it is one of the important sessions for people? Priya, the meeting is getting recorded. So you will get the recording. So no need to place another request.
Okay.
Okay, thank you. So we are on risk. So why I say that it is one of the important sessions for PBRER is because if you see PBRER, so it is periodic benefit risk evaluation report. So basically, there are two main components of a PBRER. One is benefit and one is risk. So whatever PV activities we are doing to identify any new risk or the how to minimize the ongoing risk. So everything we do as a PV person, we are mainly focused on risk part. Benefit part is mostly taken care by the medical affairs or the, you can say the medics and all those. We do draft in certain organizations. We do draft that section. You will also get to know how those sections are draft, everything. But being a PV person, our main responsibility lies with the risk of the PBRER. So in this session, I'm going to cover the risk as to you can say what are the different type of risks, how do we identify those risks, or what are the parameters that we check when we are identifying the risks to incorporate into the PBRER, and whatever it comes around. There is also a part of risk that is characterization. That is one of the important section 16.4 of PBRER. But that characterization we will cover later on because it is itself a vast topic in its own. So first in this session, we will try to see as to what are the risks, what is the classification, and how do we identify those risks as to which risk will fall under which category and how we will incorporate into the PBRER. Okay, so to start with, I hope, as I mentioned yesterday in the WhatsApp group, that please go through the section, GVP module 5 and GVP module 9. 5 is for risk management plan and 9 is for signal. So if I hope you could have gone through, but I'm not confirmed. So if you have gone through that GVP module, then you could have encountered the term risk and its different classification like important, identified, important, potential, all those things. So I hope you people went through those GVP module. If you could have gone through that, then today it would be much easier to correlate. If you have not gone through that, then also no problem. I'll teach you as to step by step so that all the concepts are into you can get it easily. So let's just start with what is a risk. So just a minute. Let me share my screen. So people do let me know if you are able to see my screen.
Yes, it is visible.
Yeah. So let's start. Risk. So risk is basically, as you people, most of you people are working into the ICSR team. So you would have encountered, means you must be familiar with the term ADR, that is adverse drug reaction. It is a common question in ICSR people when we are fresher, when we are entering into ICSR, we get an interview question that what is the difference between AE and ADR, right? So you people are familiar with the term ADR. So basically ADR, you can say that it has a susceptible causality, means the event has a susceptible causality with the suspect drug, right? Means you are not confirmed whether there is a causality associated between the event and the drug or the drug event pair. So but you are also not confirmed that it is not related. So you are in suspicion. You have a doubt in mind. It might be related to the drug. So that becomes an ADR, and that is a part of your RSIs also, whether it is SMPC or CDS and everything. So this term you are familiar that it is an ADR. Now, what is a risk? So risk is nothing, but it is a broader class, means you can say a umbrella term for ADR. ADR comes under risk. So risk is also, you can say, risk is categorized into different classification based on this causality and everything. So if suppose, first of all, understand that risk is definitely, it has either it is a confirmed causality or it has having a susceptible causality, or there might be some knowledge gap. So these are the things, the events that comes under risk, and they are definitely, means you can say that they are mostly associated with your product. That's why they are into your RSIs. Okay. ADRs might not be in the RSI, but risk will always be into the RSI. Remember that concept. Okay. So when we come to risk, did you people get my point? What I told you, how you can say that this is a risk, because it is always a part of your RSI. Remember, RSI means the reference safety information from which you pick the listedness or expectations of the product, right? So risk will always be the part of your RSI, and it is always susceptible to be associated with your drug, the drug which you are studying. Did you get my point, people? Risk.
Yes. Amit, could you please repeat once again?
Yeah. So you people are aware of the term ADR, adverse drug reactions. Those have a definition that the suspect causality is suspected into that. It is not clear. So it is suspected into that. So ADRs might be a part of your RSI, the reference safety information, like SMPC, like IB, like CCDS. But risk is always considered as, means risk is always considered as related to your drug, be it susceptible, means it might be doubtful, it might not be doubtful, it will be confirmed, it has some knowledge gap. You will understand as you understand, means if you are trying to understand the risk as a whole right now, you won't get, but if when we are going into the classification, then you will understand it more clearly. So risk, in short, you understand that it is a part of your RSI, means it is considered to be happening with your drug, and it is a part of your RSI. So when it is a part of your RSI as a risk, then there will be some minimization criteria. There are different other things that you will understand later on. So in a short form, I'm telling you what is risk. There is no, as such, no, you can say a set definition for risk in any of the modules. Okay. So it is for your understanding purpose, because people will confuse you when you will give interview. People will confuse you with signal and risk. Not, some might say that what is the difference between ADR and risk, but that doesn't come every time. It comes between signal and risk. Okay. So did you get the point? What is risk? Am I able to convey what I'm trying to?
So ADR is also a risk. ADR.
ADR is also, it might be a risk. You will come to know when we will classify, then you might come to know, or when we will go through any of the SMPs which we'll download in, then you will come to know that ADR might be a risk, but most of the ADRs are not a risk. Did you get my point? Means in a SMPC, if you see, there will be some 100, 120 ADRs or 90 ADRs, 100 ADRs. Out of those, risk will be only 5 or 6. Did you get my point now?
Yes, sir. So, sir, we can say that in adverse event is that when the causality is not been established. When susceptible causality is been established, we can say an ADR, and when the confirmed causality, then it become risk.
No. Don't confuse with that concept. You will come to know what are called as risk, because there are susceptible causality risk also. So don't
Okay.
come to a conclusion. Just what I'm telling you right now, just after I am telling, just after this, I'll tell you the classification of risk. Then you will come to know that, okay, this is the definition of risk. Okay. There is no such, like in signal, we learned a definition that if there is any new or known aspect of a new information, that is signal. There is a definitive definition for that signal, but for risk, there is no definitive definition that you can do from the classification of the risk. Okay. Am I clear?
Okay, Amit.
Yes,
Okay.
sir.
Yes,
Okay.
Amit.
Okay, so now park your questions. Let's go to the classification and then you will understand what is the risk. So let's first see what is the classification of the risk. Anybody, can you tell me what is the classification means? What are the broader categories of risks if you have gone through GVP module and all?
It's identified risk, potential risk.
Okay. Any other?
Identified, it was identified, it was potential. Confirmed.
No, there is no such term.
Important.
Missing information.
Missing information, right? Okay. So first, let's understand the concept. What is identified risk? What is potential risk? And what is missing information? Missing information we will cover after completing all this, because this is a different part. So let's come to the identified risk. Now see, what is the identified risk? As you see the term identified, that it means that it is already identified. Means based on what basis? First understand what basis we say whether it is an identified risk or it is a potential risk. Okay. So before moving to identified or potential, let me tell you one more thing, because these are the concepts which will confuse you later. So there is evidence. Okay. Evidence means any supporting evidence, any supporting thing that supports your hypothesis, that is called evidence. Okay. So evidence can be two things. Either it could be, you can say scientific evidence, or you can say confirmed evidence or sufficient evidence. So these terms you will come frequently. So try to understand that in terms of risk, what do we understand by evidence is scientific evidence means there is evidence into the literature, there is evidence into certain peer journals or all those things for an adverse event, and drug means a drug event pair combination that it has to happen. It happens with, I means just take an example, like suppose if your drug is apixaben, say, and you can say that apixaben and thrombocytopenia is the event with that. So there are many literatures, or two literatures you can say, or means there are one or two literatures that report that apixaben used to cause thrombocytopenia. So that is a scientific evidence, means someone has worked somewhere, they have done some research and they have published that paper saying that thrombocytopenia is associated with apixaben as per the researcher. So we have those
Like
articles.
it is labeled.
Not labeled. Labeled if it is in your RSI. I'm talking about scientific evidence. That is literature. If it is labeled, if it is into the apixaben RSI, whether it is SMPC or CDS, the RSI which you refer for labeling. Okay.
So that is also based on scientific evidence. Right?
We will come to that later on. But first understand what I'm trying to tell you. Scientific evidence here, I mean, it is into the literature, not into your RSI. So you can say that is a scientific evidence. Okay.
So,
If it is into your RSI, then it is either it is a ADR or it is a risk. So that you know, no issues with that. But if it is not in your RSI and it is into the literature, then it is a scientific evidence. It is not your risk. It means not a risk for the apixaben which you are talking about, right?
So it might be risk for other drug, but it
Yeah.
is a evidence of that risk, right?
It might be a risk for other drug if it is present into that other drug's RSI. But if it is not into the RSI, then it is not a risk. It is just a scientific evidence for you. Means you have some literature articles available in which thrombocytopenia is reported with apixaben, but it is not in your RSI, so it is not a risk for you. It is a scientific evidence for
Okay.
that drug event. Okay.
Amit, I have a question here. For scientific evidence, how many literature we should be able to provide to say that there isn't scientific evidence? Even the single literature article should be okay?
So in this case, when we do the signal evaluation, at that time, it is a collaborative evidence that we seek, not only the literature. We do seek our database evidence, we do seek our literature evidence, we do seek our non-clinical evidence, we do seek other labels also. So there is a collaborative evidence that we collect based on which we classify that signal that no, now it is a risk for us. So that signal becomes risk for us. So not only the literature. Literature is definitely a you can say one of the main component in deciding a signal to risk, but it is not the only component to decide.
Okay, got it. Good.
Okay. Okay. So did you get my concept? What I'm trying to tell you, that scientific evidence, I'm saying that risk means that event is not in your RSI yet, but there are certain articles available for that. So that is a scientific evidence. Okay. Now, if we see sufficient evidence, the second concept, sufficient evidence. So sufficient evidence, when we say sufficient evidence, that when we are trying to confirm that signal into a risk. Okay. So we say that we have sufficient evidence to prove that there is a causality related with the drug, between the drug event pair, and now we are classifying into a risk into our section, means RSI. Okay, so this sufficient evidence means include, just I told you, like it will be your database, whatever cases are there in your database, be it from clinical trial, be it from post-marketing. Then you will see the literature articles, how many literature articles are there, and what is the strength of evidence in that literature articles. It doesn't mean that if a person has reported for 1 or 2 patients, the thrombocytopenia happening with apixaban for 1 or 2 patients, and it will become a strong scientific evidence. There is other criteria for literature strength of evidence also. So that we will discuss when we will discuss the section 11 of PBRER at the time of writing the literature section. So did you get my point? There is sufficient evidence that in database also you find the cases. There are literature also in which there is a case of thrombocytopenia and apixaban. There are other labels, means apixaban class of drugs are there. So apixaban is mainly used for bleeding. So there is warfarin and every other class of that drugs. So in those class of drugs also, it is reported thrombocytopenia. Other than that, apixaban, if it is an established product, then there would be other MAH also who are working on apixaban or have the MAH for apixaban. So their label says that they have thrombocytopenia reported. So these are collective evidences, and then we will see the US FAERS data, ultravigilance data, in which there will be cases reported for thrombocytopenia and apixaban. So based on all this collective analysis and everything, we will say that now we have a sufficient evidence to confirm the causality. So these evidence are used to confirm the causality. Till now, you would have heard the term for ADR susceptible causality, but when it is confirmed, so it is confirmed now when you have sufficient evidence. We have evidence from all the sources to say that that causality is definitely with our product, or it is related to our product, you can say. Now, did you get the concept, scientific evidence and sufficient evidence?
Yes.
Any doubt in this or any questions? If you're not getting, do let me know. We will move slowly for this topic because you need to understand this topic in depth, because every aggregate report interview you will face, you will come with this concept of risk. What is the definition of all this risk? How do you identify? Whatever we will cover in this topic today.
Hello, Amit sir. Can you explain in detail more about this scientific evidence and sufficient evidence? What will be the difference? What will be the major criteria?
Yeah. So major criteria is, see, what are the sources for the, first of all, you understand that we have some signal and we have to make it we have to evaluate it, that signal. Okay. So whatever we will evaluate, and if it is confirmed that this signal is associated, signal means a event, thrombocytopenia, apixaban, suppose example for that. So now when we evaluate that signal, thrombocytopenia with apixaban, so if it is confirmed, then it will become a risk. Okay. So now how it will be confirmed? So when we are evaluating the thrombocytopenia and apixaban signal, at that time we will utilize different sources. as you came across the sources when you were going through the sections introduction and all those things, what are the sources? So there will be different sources to evaluate that risk. So what could be those sources as per you? Okay, let me tell. Like there will be some preclinical trials or animal studies that were done, right? That could be a source for you. Am I correct?
Yes.
Just be with me, see yes or no, okay?
Yes.
Then there would be, after preclinical, there would be some clinical trial done for that molecule before launching into the molecule market, right? So
Yes.
clinical trial will be your source, clinical trial cases, right? Then
Yes.
when your drug is into the market, there will be the post-marketing cases that will be your source, right?
Yes.
Yes.
Right? Then there will be literature. You do the literature search, means we do literature search for signals, aggregates. You get the literature cases for single patient for ICSR, right? Literature will be the source,
Yes.
right? Now, there would be labels, means suppose if you are working on a molecule apixaban. So you are a Pfizer company, suppose you are working on apixaban. Now there would be Novartis, there would be AstraZeneca, Sanofi, all these MAH are also there who are having apixaban as their molecule. They are also MAH, right? So they would also be having their RSIs for apixaban. They will not rely on your RSI being Pfizer. They will not refer Pfizer's RSI, right? They will have their own RSI. Correct, Tanmay?
Yes.
Yes.
Yeah. So in their RSIs also, their RSIs will also be a source for you. You will look into those RSIs, whether thrombocytopenia is labeled into those RSIs. That could be one source, right?
Yes.
Yes, sir.
Now, when being a MAH, you are databasing the cases. So you are submitting the cases to regulatory authority. If it is in Europe, then you will submit to Vigilance. If it is in US, you will submit to FAIRS. If you are in some RO countries, you will submit to WHO VG-based database. So their regulatory authority database also you submit the cases, right?
Yeah.
So you will screen the regulatory authority database also for the thrombocytopenia and apixaban cases. So the regulatory authority database will also be your source. Okay.
Okay.
So now you got these many sources. I told you around 6 to 7 sources from where
Yes.
you will collect the information. And now you will analyze those information and you come to a conclusion from those information that yes, thrombocytopenia is reported with apixaban. It is having a confirmed causality, means there is sufficient evidence from all these sources you took, whatever sources you took and analyzed, you came to know that there is a confirmed causality with this, between this drug event pair. So there is a sufficient evidence to confirm the causality between apixaban and thrombocytopenia. Did you get my point right now?
Yes, Amit sir. And can this all also be considered as a scientific evidence, which is preclinical, clinical, post-marketing, and
No,
all?
those are your studies. Those are for you as an MAH. But whatever is from the literature or what is means scientific evidence means whatever is reported into the literature, you can say. Okay, means there is some literature saying that there is a apixaban thrombocytopenia event, or there might be some literature only stating that there is a mechanism of action plausibility. But the term you would be coming across most of the time, you have seen biological plausibility. So that is, there is some literature saying that thrombocytopenia might happen with apixaban due to this pathway of mechanism through which it acts. So that is biological problem. So these are the scientific evidence. So scientific evidence basically you get from the literature.
Do we have any other scientific evidence rather than literature?
No, any other scientific evidence won't be there because scientific evidence is through the literature only, from the peer journals and all those things. Otherwise, we won't say that is a scientific evidence. Okay. If you get something on social media, that is not a scientific evidence. That is just an AE for you.
Okay.
Okay. Is the concept clear for you all?
Yes, Amit sir.
Yes.
Okay. So let's move now. So identified. So identified, first remember that what is the difference? This is causality confirmed. Okay. Based on sufficient evidence available. Did you get my point? Identified risks are those risks in which the causality is confirmed based on the sufficient evidence available. Okay.
So this
Now what?
causality we have to confirm, or we'll take some help for that.
No, you don't have to confirm causality here. What you have to do, you have, being a PBRER writer, I'm saying, I'm not telling you as per the signal evaluation report, because we haven't done that. So we are discussing PBRER here. So you will go to your RSI, and in that you will try to identify which is having a confirmed causality, the risk which is listed there, which is having a confirmed causality and which is having a susceptible causality. So I'll tell you how to identify all those things, but this is from your RSI that you're trying to identify. Okay?
Okay.
Okay. And potential is susceptible causality. Okay. Did you get my point, guys? Susceptible causality. Missing information we will discuss later, but just for your knowledge, it is a knowledge gap. We will discuss it later on after completing this identified and potential. So susceptible causality based on scientific evidence. Are you getting the point, guys? Identified and potential.
Yes.
Any questions here? No, right?
Amit, just for clarification, one point from my side. So we have gone through the sources like preclinical, clinical, regulatory authority, literature, post-marketing studies. So likewise, if we are getting evidence, then evidence in literature that will be called as scientific evidence, and rest of all other will be subsumed into sufficient evidence. Is that correct?
Yeah, collectively they will call as sufficient evidence, but in case if we
Sufficient
say
evidence. Yes.
if we say scientific evidence, then we refer to the peer journals, the peer articles that we have. So those are the scientific evidence. Okay.
Okay. Got it.
Any other question? Okay. So now let's understand, whenever you will go through a PBRER, you will have a concept of important identified risk and important potential risk. Okay. Have you people seen this? Any PBRER you have seen from the open source or anywhere? Have you encountered these terms, important identified risk and important potential risk? No. Okay. So we'll discuss how this is called as important. So first, let me know, are you people able to get the concept what is identified and what is potential risk?
Yes.
Okay.
Yes.
So
Yes.
yeah. So potential risk is the causality is not confirmed here. It is susceptible. Okay. And the identified risk is the causality is confirmed. But remember that these will be there in your RSIs because you are identifying from RSIs, right? This is
Yes,
nothing
sir.
new. Like in ICSR, you will get some ADRs, some AEs, new AE, new ADRs. But these are whatever risk I am telling you, these are already there in your RSI, from where you are trying to classify them into important identified or important potential or missing. Did you get the concept? Somebody might confuse you. That suppose you get an ICSR case of thrombocytopenia and apixaban. So how will you classify it into a risk? Means what risk classification you will give it. So don't tell them that this is a risk or something. Say that we don't identify from ICSR cases. We see the RSI, whatever risks are listed there, based on their criteria, we try to identify whether it is an important identified, important potential, or it is a missing information. Okay, don't confuse with that. People might check you whether you are just done it theoretically or you know the things. So whatever risk we are classifying here, it is already present into your RSI from where you will classify those. Okay. Okay, people.
Yes,
Okay.
sir.
Just be with me because I need to understand whether you are getting the concept or not. Okay. So important identified risk, important potential risk. Now we will come. So why it is called as important? Okay. Identified risk, potential risk. The definitions remains the same. But why it is called as important? For important identified risk, there is a that important. Why it is called as important? Because it have an impact.
It will have impact on risk benefit ratio.
Correct. Benefit risk profile. That is why it is called as important identified risk. Okay. Did you get my point? Why do we call it as important?
Same for potential, right?
Same for potential also. It has the benefit impact on the same thing, have an impact on benefit risk profile. How can you say that there is an impact on benefit risk profile? Anybody? How can you say that there is an impact on benefit risk profile or benefit risk ratio?
By assessing the benefit risk evaluation, means like if risk benefit of that drug is more, we can consider it as that.
So what are the parameters that you will look when you are assessing a benefit risk profile? I'm just telling you for your knowledge for the current time, but when we will discuss benefit risk, then we will discuss it in detail. Just for your knowledge, what could be
Suppose, with an example, we can say that suppose that drug is used for an oncology or for cancer, and if it is causing an hair fall or alopecia, then we can say that benefit is more than a risk.
Okay, so I get your concept, whatever you are trying to tell. So just I'll let you know certain things. See, whatever I'll tell you, which you can understand in your terms. Okay. So there is a characterization of risk. I told you 16.4 that is done. Okay. So in those, there are certain topics like seriousness of the event. Okay. Yeah, risk. You can say now, don't say event. Seriousness of the risk. Okay. Then frequency of the risk. Then public health impact. Then reversibility. So these are few parameters that I'm trying to tell you. I'm not confirming everything right now because you will confuse. So these are the main parameters you can see. So what do you mean by seriousness? Suppose if there is some cardiac disorder that is life-threatening event that is having an impact, means the patient can die. So that is the seriousness. And if it is simply suppose nausea, vomiting, that is not that much serious. So it won't have an impact on the benefit. But if it is cardiac arrest, if it is something, so that is having a more serious. So that can have an impact on benefit. Correct? Right? Did you understand the term seriousness? Why it is there?
Yes.
Yes.
Okay. Frequency of the risk. Suppose if it is an event of, let's say, dizziness, but it is having in 1 or 2 people, no problem with that. But if it is very common, it is having in suppose 100 patients are taking and it is occurring in more than 20-25 patients, then it might have a public health impact. Okay. So it is means it is impacting a mass public and it is hampering their day. Then it will become a frequency will be a part you can say for to analyze the benefit risk profile. It is having low weightage. It doesn't means carry that weightage, but you can say that it can be one factor which we try to analyze when looking at the benefit risk profile. Then public health impact means what is the impact on the public? Like we discussed the cardiac arrest. So it is having a major impact on the public health. So it is having a benefit risk profile hampering it. Reversibility. Suppose if there is some person is having dizziness, then are we able to reverse it, or do we need to give some medical intervention to reverse it, or we can reverse it by just stopping the drug, like rashes. Rashes can be reversed by just stopping the drug. So these are all the parameters that we check when we say that whether there is an impact on benefit risk profile. Anybody could ask you, how could you can say that it is having an impact on benefit? So you can say that it depends on seriousness of the risk as to how serious the risk is. How can it be reversed? What is the public health impact? Means how it is impacting the public, how population is using it and all those things. So these are the parameters based on which you can say that there is a change in benefit risk profile, or there might be a change in benefit risk profile. Did you get my point?
Yes, Amit sir.
Yes.
Yes, Amit.
Okay. So I believe now you have got the concept of important identified risk and important potential risk. Okay. And a bit extent, I told you about the characterization. So there is seriousness, frequency, public health imperative, reversibility, preventability is there. There is biological plausibility, means mechanism of action. There is a strength of evidence. There are 7 to 8 topics based on which we characterize the risk. Okay. So this is just a glimpse of characterization. These are just 3 to 4 parameters that I told you. So till here, are you guys are with me, or you are having any doubt? So let me know so that we can move further. Any
No
doubt,
doubt.
people?
No questions.
No questions. Okay.
No, Amit.
Okay. So now let's move on to understand what is the missing information. I told here that it is a knowledge gap. Okay. So knowledge gap, it is clearly evident that there would be some gap into the knowledge of that risk that you are characterizing as missing information. You don't have the evidence, or you don't have studies for those. So for example, suppose if we conduct a clinical trial for a new molecule, suppose I am conducting a clinical trial for, as one of the person mentioned, for oncology drug. I am conducting a clinical trial for oncology drug. Okay. So now when I'm conducting a clinical trial, there are certain special populations that we exclude from the trials if the drug is not intended for them. By this, I mean that suppose I am conducting a trial for oncology drug, but it is not intended to be used into the pregnant woman. So I'll exclude the pregnant woman from the clinical trials. Okay, they will be under my exclusion criteria. I will exclude the pregnant woman from my clinical trials. Did you get my point? What is the exclusion? Means what I'm talking about, the special population. Special population, you know? You
Yes,
guys know
like
special population?
pediatric
Yes.
pregnancy.
Yeah. Pediatric pregnancy, patients having renal impairment, patient having hepatic impairment, patients having cardiac history. So these are the special populations. So because they already have certain means important diseases along with them, or certain important condition with them, in which if we use drug, they are more susceptible to adverse events. So that's why we consider them as a special population and keep them aside in clinical trial. Most of the clinical trials, if they are not intended, means if the drug is not intended to be used in, suppose in pediatric population, then pediatric population will be excluded from the clinical trial. So these special population are always, most of the times, excluded from the clinical trials when we are conducting clinical trials for a new molecule and disease. Did you get my point, people?
Yes. Yes.
Yes, Amit.
Okay. So now, when you have not conducted a trial on those population, so definitely you don't know the safety impact of your drug on those patients, right? So when you don't know the safety impact on those patients, so that is a knowledge gap, right?
Yes.
Did you understand the concept of knowledge gap? Why I'm saying a knowledge gap? Because I have not conducted the studies on those populations, so I don't have the data. So that is a knowledge gap for me. And in the post-marketing phase, when your drug will come, you don't have control. It might be used by a pediatric patient who is having oncology condition for which the drug is into the market, right? Am I correct? Did you get my point?
Yes.
That will
Yes.
be a off-label use. No doubt that will be off-label use, but it might be used, right?
Yes.
Correct.
Yes.
So that is a knowledge gap. So now what happens? You got the concept, what we exclude into the clinical trial, what is the knowledge gap? Now, second thing, somebody can ask you, so since we are excluding these population into clinical trials, so all these will be are missing information, means use of your drug in pediatric population, in pregnancy population, in renal impairment patient, in hepatic impairment patient. So we are excluding everything into the clinical trials. So all these will be in missing information. What is your thought on that? People, speak something. Be it right, be it wrong. No problem. Speak.
Yes,
Yes.
sir.
It will be under it.
All will be missing information, right?
Yes.
Don't be afraid. Everybody is learning only. Don't be afraid that I'll be wrong or right. So all will not be your missing information. Otherwise, every drug will have all this patient population, special population by default as missing information. So now comes the point, then what will be the missing information, right?
Yes.
Yes.
Okay. So what will be the missing information we will get? You have done animal studies, right? Before launching the molecule for clinical trials or getting the approval for clinical trials, you have done the animal studies, correct?
Yes.
Okay. So in animal studies, we do toxicity studies, different type of toxicity studies, carcinogenicity studies, cardiovascular studies. We do all those studies in animals, right?
Yes.
So now are you able to correlate where we will get the missing information from?
So we'll refer to the preclinical study data.
Right. We will refer to the preclinical study data and we will see whether the testing was done in pregnant mice or pregnant animals, and was there any toxicity into that pregnant mice reported with our preclinical studies? Did we do some carcinogenicity studies or cardiovascular studies into our animal studies, and did we get any safety findings from those? If it is available in your preclinical studies, then those will become your missing information, because in animal studies you have find the toxicity, but you didn't conduct the study in those patients in human beings in clinical trials. So there is a knowledge gap and there is a possibility that this safety profile could emerge in the humans also. So we will keep it as a missing information. Suppose if it was reported that a cardiovascular study was done in the animals and the QT prolongation was found in the animals. So now QT prolongation is one of the missing information. It means QT prolongation could be there into the this cardiovascular patients. So you will keep use of drugs, your use of your molecule जो भी use in cardiovascular history patients or cardiovascular patients. So this will be your topic of the missing information, use in cardiovascular patients, yeah, cardiac impaired patients. So this will be your topic of the missing information. And whenever you will get, means whenever you are analyzing this, so there will be search criteria that we will discuss when we are discussing 16.3. So you will try to identify those cases in which the patient is having a medical history of cardiac disorder, and if there is any QT prolongation event reported into that. So that will start confirming your hypothesis that QT prolongation could happen in a cardiovascular impaired patient. So that's why it is my missing information. Did you get my concept? What I tried to tell you here? Missing information concept?
Yes.
Yes, sir.
Any doubt, people? If you're not able to understand, do let me know. I'll explain it again.
Yes, Amit sir. So, like for the missing information, so we will try to get information from the like ICSR or the clinical data.
No. For missing information, we will not see into ICSR, nothing.
Okay.
See, forget the ICSRs and everything. Missing information is from your RSI. This risk, whatever important identified, important potential, missing information, we are taking into from your RSI. Okay. So missing information, the preclinical trial, you will see the animal study data that will be present in your RSI. And if there is any safety findings reported in any of the patient population in the animal studies. Did you get the concept? Don't
Yes,
confuse
yes.
with ICSR.
Yeah.
Don't confuse with other
Okay.
evidence. That was for your understanding. Now only remember that you are having your SMPC or RSI in front of you and you are trying to identify this risk from that RSI. Simple.
Okay, right. Yes.
Okay. So for missing information, you will means you will check your animal studies data. If there is any safety finding in any of the animal group, whatever is the
So
dump.
what if the preclinical study data is not there for
So
that
no missing
particular?
information. No missing information. Missing information will be none.
Okay.
Okay. Any other doubt till here?
No doubt, I mean.
People, anybody?
Yeah, Amit,
It is clear, Amit.
apart from this, actually, any other scenarios are there? Like, if we consider that this is a missing information, like you told, like RSI, if you are checking the RSI, if there is nothing there reported, and if you are cross-checking with the preclinical data, if something is there, then we can consider it as a missing information. So, based upon the tolerability or toxicity level, if any, for the specific population or pregnant. So that I understood, but if anything else apart from this, like if that is not there in that RSI, and if it even if it is not required for the preclinical studies in that, and we can still consider it as a missing information. Is there anything apart from this?
See, if you talk about risk that has to come into PBRER, then it has to be into your RSI. That is the thumb rule. You can't go beyond that. It has to be there in your RSI. If it is not in RSI, then it is not a part of your risk in any of the risks, whether it is important, identified, potential, or missing. It has to be in your RSI. Okay?
Okay, understood.
Because that is your standpoint in terms of that is your company standpoint for regulatory authority. So whatever is there in your RSI, that only will become a risk, whatever risk it will become, in whatever way you can classify it. Okay?
Yes, sir.
Any other question, people?
No.
Shall I move ahead?
Yes.
Yes, Amit.
Yes, sir.
No question, people, right? Why am
No
I
question,
asking
Amit.
every time?
No.
Because people get confused. After this class, you will be confused, I'm sure.
Sir, I had one question.
Mhm.
So you said for missing information. So for everything we have to consider, we have to see the RSI. Okay.
Mhm.
So to identify the missing information from the RSI, we will see the animal section in the RSI. If it is
Right.
if it is something is mentioned in the animal section, we will write it with the missing information. Okay.
Mhm.
And about important means like identified risk and potential risk. So for this, which section we will refer to the RSI, means like we know that what is important identified risk, and we know that what is the potential risk, and we know that these are the classification based on the evidence. So but in RSI, which section, or is it written in the RSI only that these are the uh, in means like identified risk or potential risk?
Mhm. So this is the next part that we are going to cover. So till now, if it is everything is clear, then we will go to cover as to how we identify this risk
Okay,
from the
okay.
RSI, or from what are the sources, how we identify this risk.
Hello.
Sir,
So
in RSI there is a means like section where it will be mentioned like risk is risk, or it will be mentioned under ADR.
no, RSI, when you go to as SMPC, there are three main sections. That is 4.3 contraindication, that is 4.4 special warnings and precautions, and then section 4.08, that is ADRs. So from these sections, you try to identify as to what are the risks that I have to include in my PBRER and in which class they will fall into.
Okay, okay.
And for missing, you will see the animal study data.
Animals. That is clear. Okay.
Okay.
Apart from animal study, is there any other we can see evidence for missing information?
No, only animal studies. Because knowledge gap is you have not done the clinical trials and all those things. So what you have done the animal studies. So that is the only source from where you can get the missing information.
Okay.
Any other question?
No, sir.
Great. So now let's move on to as to how we can identify the risk. Okay. Chalo. Now let's do some internet work. Tell me some molecule name. Anybody? Tell me some molecule name. Newer ones, not the warfarin and all those things. Anyone?
Adalimumab.
Adalimumab. Chalo. Adalimumab. Okay. First of all, before searching, I'll let you know certain things. So when you are going to write a PBRER, so when writing a PBRER, so I am referring here that you are writing the first PBRER. Always remember there will be questions in the interview regarding the first PBRER. Because if you are doing second PBRER, then you can take risk from first PBRER, right? So they will ask you, suppose if you're doing first PBRER, then how will you take the risk? So I'll tell you the process, how to take risk when you are writing a first PBRER. Okay. So first thing what you will do, like suppose we are writing a PBRER for adalimumab. So to see the risk, first we will see whether we have an RMP for that adalimumab. RRMP means suppose I am a Pfizer company working on it, or adalimumab is basically, you can say, AbbVie's product. So AbbVie, you can say, suppose AbbVie is having an RMP for adalimumab. So if there is an RMP for adalimumab, then you can take risk from there directly. One thing, right? Did you get my point? First, how do we search?
So we take that section as it is from RMP.
Yeah, the safety concerns means
Okay.
the important identified, what are the important identified risks, what are the important potential risks, what are the missing information in that RMP. Okay.
For our company or for any company?
For our company. First, for our company. First, always you will go for your company. Your company molecule, adalimumab, you are writing first PBRER for adalimumab. So you will first search your company's adalimumab RMP into your internal database. There will be, if it is RMP, then in the RMP part, it will be there, always there. Adalimumab will be there. But it might happen that your company do not have an RMP. It happens mostly in the established product case. So we will go step by step as to how we search for the these risk. Okay. So first understand that you go to your RMP and see whatever the risks are listed there. So you will take those risks, right? First thing, first step. Okay.
Yes, sir.
Okay. Now here comes another question. Some people might ask, okay, then whatever risks are there in RMP, you will take those. So does you faced any scenario in which the risk were in the PBRER, risk were more than the RMP or included certain other risk also, which were not there in RMP? So what you will see? If I'm asking this question, then there would definitely be this scenario, right? Like suppose. What happens? Just a minute.
Hello.
Just a minute.
Okay, okay. I thought issues.
So what happens that suppose if you are writing a PBRER for a European region. So in European region, we basically refer SMPC as an RSI, right? So RMP means I will not give you more complex details, but just try to understand that if there is any additional risk minimization activity or any additional PV activity ongoing for a risk, then only it is included into the RMP. Additional risk minimization activity, ARMM, or additional PV activity. then only it is included into the RMP. Did you get my point? Did you people, I hope you people won't be having the concept of additional risk minimization and additional PV activity, right?
No, we don't.
That's why.
No.
That's why I am telling that I will not give you much complex. Just for your understanding, just get this point that if there is any risk, means it is classified into important, identified, important, potential, then there will be a risk minimization plan for that. That's why RMP is there. Okay. So there will be risk minimization activity. So there are two types of risk minimization activity. One is routine, one is additional. Routine is that will be available in your RSI as to suppose if some your risk is hepatic disorder. Okay. So your RSI will say that monitor the patient for their AST, ALT level. If it is above this, then don't administer this drug. So this is a routine risk minimization that is already there in your RSI. Okay. Did you get my point? What is routine risk minimization?
Correct.
Now, additional risk minimization. Now, additional risk minimization, they will say that will not be a part of your RSI. Okay. It is not necessary to be a part of your RSI. That will be a part of your RMP. Now, suppose if a drug like we had teratogenicity with the thalidomide, you know, but still we have those class of drugs like pomalidomide, lenalidomide. These are being still marketed into, and there are blockbuster drugs for oncology and all. They are still into the market, right? Now, teratogenicity is the risk associated with the pregnant woman, correct? Means a pregnant lady took the thalidomide drug and their fetus or their babies were born with distorted limbs. So that was teratogenicity, correct? Did you get my point?
Yes.
Now, suppose lenalidomide is into the market right now. So what should I do so that this teratogenicity risk is not evolving into the market? So what can I do? I'll restrict the use of drug into the pregnant woman. I'll plan my risk minimization that this drug should not be used by a woman who is going to be pregnant or who is susceptible to be pregnant or who is pregnant at the time of starting the drug. Did you get my point? People.
Yes.
Is it exhaustive? Is it getting exhaustive?
No.
No, sir.
It's fine, right? You are with me. All of you are with me, right?
Yes.
So now, when I have to plan that it has not to be given to the pregnant woman or the woman who's going to be pregnant. So I'll have a plan. So what plan could be there? Like I'll circulate some healthcare professional letters to the doctors who are prescribing, or I can enroll the doctors into my, means I can restrict the distribution of the drugs. So I'll enroll the doctors, pharmacists, the patients into my database. And from there, the drugs will be circulated to the patient so that it is in my control. There will be a restricted access to the molecule. So these are the additional risk minimization activities you do to minimize the risk of teratogenicity. So did you get the concept of additional risk minimization with an example I told you?
Yes,
So
sir.
obviously this cannot be included in RSI. That's why it is additional, right?
Yeah, that is apart from the RSI that you are doing. So, but it would be there in your RMP. This additional risk minimization activities you can find in your RMP. And in PBRER, what will happen? There is a section 16.5, effectiveness of risk minimization. So there you will tell as to how effective the risk minimization. It is not that it will not be there in your RSI. It will not be there in your RSI, but in PBRER, it will form your part. In 16.5, it will come to be as a part of their PBRER, that what risk minimization activity you are doing and what is the effectiveness of that risk. Did you get my point?
Correct. But sir, what is the what is the need of it? Means when we are saying that we have mentioned in our RSI only, we should not administer this drug to the pregnant woman.
Then also people used to take it. Off-label use concept, you know, na? You get the cases for off-label use. So people tend to use that. But we don't want that to be used as off-label also in pregnant ladies. So that's why we are doing the additional risk minimization, because it is teratogenicity is the high risk due to which PV itself came into existence. right? So we have to control. Being an MH is that it is our responsibility to control that it is not being used in the pregnant lady. We can't say that we have mentioned in the RSI, so we are devoid of it. Now, no need from us to monitor it. It is your responsibility. Your drug is into the market. You being a MH, you have to monitor and you have to plan the risk minimization activities and everything. Okay?
Okay. Hello.
Yeah, tell me.
Hello. Yeah. As you said that routine RMP is a part of RSI and additional
Routine risk
RMP.
minimization. Routine risk
Yeah.
minimization.
Yeah. And additional risk management is not a part of RSI. So
Right.
as you take an example for the thalidomide.
Mhm.
So in the thalidomide, RSI is also involved that line that this should not be administered by the pregnant woman. So it is also part of RSI. So how we can consider it as an additional risk management? If it is a if any marketing authorization holder mention in their RSI like it is not administered for the pregnant woman, then how we can consider it as an additional
Additional RMM, right?
yes, correct.
Yeah, so don't confuse with all those. RMM is a different part. Okay, so routine, when I say, so you correctly mentioned that, and you got the concept also that it will be there in your RSI that don't use for pregnant ladies. So this is a routine. Routine means it is there in my RSI. It is a routine. It is a part of prescriber information. The prescriber will know. But what is ARMM now? You say so. ARMM is I am having giving a restricted access. I have planned a distribution program. So that distribution program is an ARMM for me. That is a risk minimization activity for me. Okay. Risk minimization measure you can see for me. That distribution program, the restricted distribution program being conducted by me as an MH is an ARMM for me. That will not be a part of RSC. That will be a part of my RMP.
Okay.
RMP
Got
is
it.
a risk management plan. So what risk management plan you have? Suppose here teratogenicity is the risk. So what is the plan that you are having to manage this risk or minimize this risk? So that plan will be a part of my RMP and that is an additional RMM. Okay.
So routine and additional both will be in risk management plan, right?
Routine is there part of our RSI. That is not much important because that is a part of our RSI. So forget about that. Additional RMM is most important for any risk.
Yeah, but both will be included in RMP.
Yes, they will be there in RMP.
Yes.
But in RSI, ARMM will not be there. Don't confuse, okay?
Okay.
Any other question, guys? Actually, risk is a vast topic. We dig into it, it will. Okay, let's move on to additional PV activities. Just in short, okay? I discuss the concept of characterization of risk with you, section 16.4 into PBRER. So additional PV activities. Now routine PV activities and additional PV. So routine PV activities are those that we are doing. Like we have a safety monitoring program like ICSR management and all those things are there, right? We are doing routine signal detection activities. So these are the routine PV activities. We are having, suppose you could be having some questionnaire or follow-up questionnaire for certain molecules, drug event pair. Am I correct? Like drug-induced liver injury is there with some molecule, then there would be some targeted follow-up questionnaire that you process into your ICSR as a follow-up, correct? People?
Yes.
So these are routine PV activities. The ICSR cases that you are getting, the signal detection activities that you are doing, the follow-up questionnaires that you are sending, these are the routine PV activities that you are doing. But what is the additional PV activity? So additional PV activities are the characterization I mentioned. So when a risk is identified at that time, the risk might not be characterized because you don't have the sources or you don't have the evidence to characterize it completely. So whatever characterization parameters are missing or whatever characterization topics you are missing into that. So to fulfill those topics, we plan a post-authorization safety studies. So if we plan a post-authorization safety studies, then those safety studies are called as additional PV activities, just for your understanding. Just don't go deep into this. We will study when we are doing characterization and all. Did you get the point? What is the additional PV activity and what is routine PV activity?
Yes, Amit.
Yes, sir.
Any questions, people?
No, Amit.
Okay then. So where were we as to like one of the participants asked whether RMP risk we will take directly. So this happens here. So in RMP, there will be the risk mainly, which is having a additional PV activity or ARMM ongoing. So those will be there mostly. So that's why in PBRER, there might happen that there are more risks, whatever is present in RMP. So from where those more risks will come, like I told, we are doing European PBRER. So we will refer SMPC as an RSI. So in SMPC, there might be certain more risks listed. So as per the region wise, so we will take those risks for European PBRER. So there will be additional risks apart from the RMP, correct?
Yes.
Yes.
Any other question, guys?
So RMP includes additional studies we are doing. And if
No.
apart from that, we are
RMP
taking from
RMP include those risks in which either ARMM or additional PV activities are ongoing.
yes. Apart from that, we'll refer the RSI of that particular region.
Right. Or if it is a global PBRER, then you will refer CDS.
Okay.
Okay. So first concept covered that we will go to RMP, RRMP and see if there is any RMP available so that we can take risks from them. Okay. Okay. One more thing, understand that it is this risk identification is our responsibility, but the accountability lies with the safety physician. So whenever you enlist this risk and you present it to the QM1, we discussed kickoff meeting at the time of planning of aggregate report. We do conduct a QM at DLP minus 25, I think, or 60, whatever is there. So we conduct that kickoff meeting. In that kickoff meeting, we enlist this risk and get it confirmed with the physician. So accountability is with the physician. So if somebody asks you, somebody can confuse you that, okay, so you have enlisted the risk and that is final. So you can say that, no, we enlist the risk, but it is finalized in the kickoff meeting in agreement with the physician of the molecule. Okay. Don't take it everything on you. We are just being a writer, we are just presenting all those information, but everything lies with the physician, whatever key concepts are there related to risk and benefit and all those. Okay. Let's move forward, right? Now,
Yes.
suppose adalimumab is now into the market, its patent has expired. So there are many MAH for it. So it might happen that being, suppose you are a small company, like say, Zen Pharma, Den Pharma, any pharma, you are marketing adalimumab. So you might not have the RMP for adalimumab, right? Because since you are a generic company, you are marketing the adalimumab generic product. So you might not have the RMP. It is not necessary that you have the RMP. So now where will you get the RMP? So we will search as to where we will get the RMP. It's for our easiness that from the past October month, EMA has mandated it to have at least RMP summary onto their website from all the MAH. So we get the RMPs now easily from there. So EPAR, remember, right? EPAR, European Public Assessment Report. Did you get the meaning of EPAR, people? European Public Assessment Report. Okay. So we'll go on this website. Minimize. Okay. Now, just search here adalimumab. Or adalimumab's brand name is, I believe, Humira, right? Humira is by
Yes.
FV. Okay, we'll search Humira. Let's see what happens. I just did human for my means just for safe use, in which was human also 54 results are there and there is 51. So we will go to see where is Humira. There are many drugs now being used. I think Humira is, this is Humira. So we will open Humira. Okay. Now Humira. EPAR medicine overview. And this is risk management plan summary. They have just given us summary. Okay. And this is product information. So this would be the SMPC. So let's first open the RMP summary. They have not given a complete RMP, but a summary is there. So let's see what is there in the summary. You can see, right? Summary of safety concerns. Module part 2 का 7 part. 7th part is summary of safety concerns, means the detail, and then summary would be in 8th part. 100, page number 100. Let's see. I'm trying to show you the procedure. Just be with me, okay? Now see. These are the summary of safety concerns listed here. Can you see?
Yes.
Can you guys see clearly? So this will be a part of your risk when you are writing a PBRER. These are the important identified risks. These are the important potential risks. And these are the missing information. Okay.
Whenever we are going to add this information in February, are we
Mhm.
going to cross-refer this particular document, or there is no need to do that?
No need to cross-refer. It
Okay.
is just for your internal use, means when you are enlisting this risk and when you are going into the kickoff meeting, then keep a reference of this document with your physician to understand to make your physician understand that from where you take the risk. And second thing, what you have to do, you saw all the risks here. Okay. So now you have listed this risk into your means QM document. First, we will talk about QM document. PBRER is the, when you start writing, then PBRER will come into picture. So in QM document, you have prepared these as your risk. Now you go to your RSI and see whether this term is there in your RSI or not, or is there some other term in your RSI. So you will modify it accordingly. If it is not there, then you can write a comment for your physician. Then it is not, it is there in the EPAR of this Humira, but in our RSI, this term is not there. So he will take a call and say that, okay, delete it from the risk, but it has to come from your physician. Okay.
Okay. Okay, got it.
Did you understand the process? Yes, tell me.
Yeah, so I'm looking at the missing information here and
Mhm.
says aged from 6 years to less than 18 years with CD. So you said that we'll look at the animal study, but how can animal study be conducted like for 6
See,
year, 18 years?
so animal studies will say that there will be some fertility issues in there. There will be fertility studies done in which babies will be born abnormal, or there would be certain, you can say in your RSI, you will find this term that this is not, means this is not, you can say this is not intended to be used into this patient population. So based on this, you will look into your animal studies. If there is any toxicity reported, so you will see that if there is some toxicity reported in fertility studies, so you can take this term. We will also always take from our RSI. That could be different, like 6 years to 18 years. This will be there in your RSI in under special populations from where they have taken it. Okay, let me clarify it. Let's look at the product information for that. What is there in the product information? Okay, now we are into the product information. See, Humira is indicated for the treatment of moderate to severe acute from 6 years of age who have had an adequate treatment. Below 6 years, it is not intended. From 6 years, they have for pediatric, and up till 18, we say that it is a pediatric. We don't consider them as an adult, right? One hint you got from here. So these are the things that when we will come to RSI identification, then I'll tell you as to how we identify the terms as to what term we have to take. Now, when you will go to the special population, see, those of our patients with Crohn's disease from 6 to 17 years of age is based on body weight. Okay, body weight is. They are saying no, Humira is not studied in these patient populations. But 6 to 18 years is coming because this is approved in that group. So it would be studied in that group. Did you get my point?
But it's in the missing information, right?
Missing information because they don't have the safety information. Most of the safety information they are not got it. So let's go to the patient more population group. Just a minute. We will see as to how that term has come. Fertility, pregnancy. See, women of childbearing pregnancy at approximately post-development resulting in with no organ putting more than 1500. You will find see the primary endpoint was the birth prevalence of major birth defects. Ending with at least one life in that father. Continuous version, minor birth defect, preterm birth size, and a serious or opportunities were reported. See, these are the toxicity reported in pregnant women. So it is studied in pregnant women also. Okay. Yes, feeding. So this drug is being used. Clinical studies. This preclinical safety. The embryo fetal development toxicity perinatal development study has been performed. Okay, very less preclinical study they have given. What is the term being used here? Long-term. Oh, see, the term is long-term safety information in the treatment of children from aged 6 years to. Now, understand the meaning of this long-term safety information. Okay, what happens when you do a clinical trial? It is for certain time phase, means 6 months, 1 year, 2 year. But we have not studied the long-term safety of that because this drug is intended to be used in this 6 years to less than 18 years. Okay, I didn't see the long-term safety term. That's why it was confused. See.
So for this, we no need to look into the animal studies,
No,
right?
no need to look into the animal studies. They have suspected that there could be some long-term effect. So that's why they are saying that long-term safety information means this long-term you need to analyze with your physician. Just you will have to look into the CSR as to what was the duration of this study, the pediatric study that was done. Suppose if it is 18 months, then you and your physician means your physician will give you the heads up that, okay, consider the use of Humira into the children between 6 years to 18 years in which it is approved. So duration he will consider, like suppose if it is used more than 36 months. So now let's see what is the safety profile involving in the use of these children between 6 years to 18 years if it is used more than 36 months. So that 36 months, more than 36 months is the long-term safety information. So if any safety profile is emerging from use in from beyond 36 months, then that will be a part of your missing information analysis. Did you get my concept? Why it is here?
Yes, yes, I got it. But you said, no, like only from animal study. We will
Animal studies, I
explicitly.
am saying, animal studies, I am saying that patient population, the special patient population, you can't take all this patient population. But if there is, if you have to take missing information, you have to go into the animal studies and see if there is any toxicity reported in any of those patient populations. Then that will be a part of missing information. Otherwise, it will not.
So, Amit, along with the animal studies, we have to see other part also.
Amit, you are not audible.
I think there is some connectivity issue with Amit. Just hold on for some time. He will rejoin.
Hello, people. Hello.
Yeah, hi.
Yes, we can
Hi,
hear you. Hi.
you are audible
Yes,
now.
we can hear.
Yeah, we can
Yeah,
hear you.
actually, there was a power cut due to which net got disconnected. So I have connected with my mobile. I believe screen would not be showing, right?
Yes,
Yes,
Now can you see it?
yes, it's
yes,
visible.
we can see it.
Okay. So where were we on long term, right? So long term, what happens if we suspect that the drug therapy has to continue more than this time limit, then we will consider it as a long-term safety information, as a missing information, and we will see if there is any safety arise impact from that long-term use. Okay, it is not with all the drugs.
So apart from long term, we will look into the animal studies.
Yeah, apart from long-term, see, we will look into the animal. If there is certain toxicity for animal studies, then only we will consider that population into the missing information.
Yes.
Long-term treatment, this episodic treatment, there will be some cyclic treatment. So these are not, we don't do this in animal studies. So these are also a part of, because safety profile might emerge in when we are using a drug for a long time. Okay.
So apart from this long term and episodic, there is no safety missing information here.
No, I don't think so. We have come across only special patient population, long-term safety. And if there is some genetic molecule, then some genetic, that is also long-term only. You can confuse, you don't need to confuse with that. Okay. Any other question? People, any other question?
No.
Now let's move on to. So what I have told you, if your RMP is available, take from RMP. Not, then go to EPAR website, search for the RMP. If it is available, then take from there. But don't take directly from EPAR. If it is your RMP, then the terms would match. But if it is from EPAR, then terms might not match. So you will have to check in your RSI as to what is given. Like serious infection is there. So you need to check. So we will go to 4.4 and check whether there is serious infection is reported or what is there. Infections is reported. They have made it as serious infections. So it's fine, no problem. Serious infection is also here. Tuberculosis is also here. So this will all be part of your. So how can we minimize this here? Tuberculosis is there. So we will learn as to how to pick from RSI. First, let's go step by step. Okay. Did you get the concept how we take the risk and everything from RMPs?
So Amit, if the serious infection is mentioned here, but we are not going to add because it is already there in RSI. Is that correct?
No. I am saying that if you are referring EPAR, so if you are referring EPAR, then it is definitely that you don't have an RMP. Means being an MEH, you don't have RMP for the molecule. First thing, right?
Right.
So on EPAR website, whatever RMP is there, so this RMP could be of different MEH, right?
Okay.
Means definitely it will be of different MEH, not your. So that MEH will be having a RSI,
Right.
RMP and RSI. So this RMP, whatever you are seeing on EPAR, these terms, serious infections, tuberculosis, this will be matching with their RSI, whatever is there in the RSI.
Okay.
In their
Okay.
RSI. Okay. And I told you that we have to take risks from our RSI. So we will match these terms with our RSI. Okay.
Got it.
And whatever
Got
the
it.
matching terms are there, we will take those terms as risk into our PBRER. Did you get my point?
Yes, got it.
Great. Any other question?
This is clear, Raman.
Okay. Let's move on to your RMP done, EPAR RMP done. Now, what if there is no RMP available for the molecule on EPAR also? Then what will you do?
We'll directly take it from RSI.
There's one more step in between. RSI is the last step we will go. So, and this is for the established molecules, remember, okay? Suppose if it is your innovator molecule, means its patent has not expired. So there won't be any MAH other than you for that molecule. So you will be having RMP also for that molecule. So you will refer for your RMP, your CDS, whatever RSI you are referring, if it is a regional PBRER like for Europe, so you will refer SMPC and RMP. If it is global PBRER, then you will refer CDS and RMP of yours. If it is an innovator molecule, the patent has not expired. Did you get my concept? Suppose there is a new molecule, like suppose I'll say adalimumab for AbbVie, when it was for AbbVie only, Humira when it has not become adalimumab, means generic has not come. So at that time, Humira RMP was there with AbbVie, Humira RSI was there with AbbVie, the CDS was also there, the SMPC also. So at that time, when the person has started writing first PBRER, so he could have referred the RMP first version and he could have referred the whatever RSI is there. Okay. But when the patent expired for Humira after 20 years and it became generic adalimumab, at that time, we refer to all these things. Did you get my point? If somebody asks you, suppose it is an innovator molecule for you, so will you go to EPAR and see? You won't find anything. EPAR, your RMP will only be there on EPAR. Am I clear?
Yes.
Right, people? If you are an innovator for that, the patent has not expired, you will be having everything at your source. Means at your internal source only. No need to go for EPAR, no need to go for anywhere. You have to refer your only. But if it is a generic or it is an established molecule, patent has expired and you don't have an RMP, then you can do all these searches. And in interviews, people look for these, means what approach you apply to find the risk. Means, yeah, they want to know whether you know about the EPAR, whether you know about the CMDH, whether you know how to take risk from RSIs. Okay. Shall I move forward?
Yes. Yes, sir.
Okay. Then let's come on to the next concept.
Yeah, hi Amit, one question.
Yeah, tell me.
Yeah, so for example, our MH is in the European region, so we will use the EPAR. For example, our MH in US FDA, then we will use the US FDA website to search this data.
No, EPAR is only for European, means if your molecule is approved in European region, you will find there.
Okay.
Suppose adalimumab. Adalimumab is not approved in European region, so you won't find EPAR for that adalimumab.
Yes.
Okay.
Yes.
If it is approved in European region for any other MAH also, not for you, then also you will find it on EPAR.
Okay.
Okay. And people might confuse you that if you are writing a paper for US, then how will you take risk? So don't confuse yourself. US, we write PADers. And if you are submitting PBRs, then it will be as per the European regulations.
Okay.
Okay. And suppose if you are only MAH in Canada region, let's suppose, where you have to submit, you are submitting PBR, then also you can refer to EPAR website and in agreement with your physician, you can finalize your risk.
Okay. Okay.
Okay. EPAR is the standard website for RMPs and SMPCs.
Yes.
Okay. So let's move. Let's move, people. Next.
Yes. Yes.
Now I am searching CMDH. See, we will go to CMDH site. We will search for this list of safety concerns. It will be in the Excel form. We will open it. It's still downloading because I am connected through mobile. Now. So meanwhile, you understand, first we look for RRMP, then we go for EPAR RMP. If RRS is not available, from EPAR RMP, we will search and match the terms with our RSI and populate accordingly and get confirmation from physician during the QM, right? Now let's move on to the CMDH. If it is not available on EPAR also, the RMP summary or RMP is not available on EPAR, then there is one more thing, that is CMDH, list of safety concerns. So this is how it will look like. So see, this is CMDH line listing. Okay. Now what will happen? You want to see adalimumab, correct? So we will see if adalimumab is available here.
So all the drug in EU region will be here.
Yeah, most of them whose RMP is there, they will be available here. If RMP is not there, then their listing will not be here. Okay. So adalimumab, they're not having adalimumab here because it is established product now. Okay, leave it. Let's look for some other. Like suppose if I go for any molecule, L-prostadil or allopurinol, suppose. These are the lists you can find here. L-prostadil. So this is just for your knowledge as to how you look into the CMDH. So see, this is the product name. This is the RMP version, number and date, and this is the safety concern. So safety concern, you will find this is important identified risk, this is important potential risk, and this is missing information. Okay. So now you will go to the latest RMP version. So like suppose if you are having, this is the 2016 dated, where I have pointed right now, you can see this is the 2016 website, right? So we will move
Yes.
further. What is the latest one? This is version 2.1. This was version 1.1. This is version 2.1. This is version 3.2 dated 2018. This is version 1.1 dated 2018. Okay, different molecule it is. I mean, it's different MAH it is. Then 2018, then 2019. This is the latest one, 2019. Okay, in all the RMP versions, this is the latest one. So we will pick the risk from here. So this is the identified risk, hypersensitivity reactions, including SJS and 10. Okay, this will be your important identified risk from here. Then this no important potential risk for this, and use in pregnant women is the missing information for this. Did you get how do we find from CMDH? People?
Yes,
Yes.
yes.
Yes, Amit.
Okay.
Amit,
So
always we will be looking for this, the updated one, right? The updated version, right?
yeah, the latest one, the most latest one.
Okay.
Yeah. There is another criteria like suppose this is 2019 version. You see your RSI as 2020 version, then it is perfect. You can use it. No problem. But if your RSI is 2018-17, then you need to match the risk terms and everything. But risk you will take from this the latest one only. Okay. You can delete some risk also. So that is a different part. Don't fall into that. But you will always refer to the latest RMP version, means date.
Sir, what is the use of CMDH when RMP is already present? You said that those are when RMP is present, only those drugs will be listed in this, means like in CMDH. So when RMP is already there, why should we go to the CMDH? Means like it is compulsory to check it, or just
No, first it is compulsory to check the RMP. If the RMP is not available, then you will come to the CMDH website, because there might be the scenarios that your molecule doesn't have an RMP in the European niche, because European region has two different procedures to get the molecules approved. There is centralized authorized procedure, there is national approval. So sometimes due to those conditions, it might happen that the RMP summary or RMP is not present or EPAR, but on CMDH website, you will find that.
okay? So
In
first
this line list.
we will see EPAR, and
Right.
if it is not there in EPAR, then we'll go to the CMDH.
Then we will. Correct. This is the third step. That's why I'm telling you step by step.
Okay.
So first step is search in our database. Second is EPAR, and third is CMDH.
In our RMP database, don't say only database, otherwise they will say Argus or ISG. In our RMP database.
Okay.
Then EPAR website, and then CMDH. Okay. And this listing gets updated. So you need to go on the website when you are using it so that you can have the current version. Okay, great.
Yes, Amit.
Yes, sir.
Okay. So this is done. Now I'll close it. So now searching the websites is done. So next comes as to how we can identify our risk from our RSIs. Let me download one SMPC then. Adalimumab only we can have now. We had last time. Sorry.
So this is after RMP, after searching in RMP.
Now after you searched RMP, you search EPAR website, you search CMDH web lines, means that Excel, but you didn't get your molecule anywhere. So now how will you pick the RSI? This is the main question they will ask you again. I mean, suppose if you don't find in all these things, then how will you pick the risk? So then your answer will be that we will pick from our RSI. So they will ask as to how you will pick from our RSI. Did you get my point? It happens in generic molecules when you are doing, or established molecules when you are doing, it happens that you don't find the RMP, you don't have your RMP, you don't find EPAR RMP, you don't find it in CMDH line listing also. So now you have to pick from your RSI. There is no other option. Okay. Shall I move ahead?
Yes.
So
Yes.
earlier you mentioned that about causality and all that. We don't have to do that. It is already done in the RSI and RMPs, right? And categorized as a risk.
In RMPs, it is always, means till now, whatever you have seen how to pick the risk, so you don't have to do the causality and all, because it is already there in all these things. If you find this thing, then it is easy for you. Okay. But if you have to pick from RSI, then you don't have the causality there. There won't be straightforward causality that it is established or something like that. There are certain parameters based on which we identify the risk and get it confirmed from the physician. Okay. Till now, if you have find, suppose if you've gone through all these 3 steps and you found the risk, so there is no headache of checking the causality and all. You can just match the terms and put them into the appropriate category as mentioned into these sources that you have seen, right? But if you don't find in these 3 sources, then it becomes a headache for you to identify the risk.
So before this, I have one question. So you said in RMP also, if you are getting any EPAR RMP, we have to refer to the RSI, and then only we have to this. So in all those above 3 steps which you said in RMP, EPAR, and CMDH, every time we have to refer RSI for that also, na?
Yeah, every time you have, suppose if it is your RMP, then also you have to refer CDS to see if there is any additional risk that can be incorporated into the PBRER. If it is from the RMP from the EPAR, then you have to refer your RSI to match the terms. And if it is CMDH, then also you have to match, go into your RSI and match the terms. But what got you as a easy or ready-made thing is the classification of the risk, so that you can put those risks into the same classified.
Got it.
Right?
Right.
But every time you have to refer your RSI to match the terms, because you can't go beyond the terms listed in your RSI. You have to keep the terms consistent with your RSI.
Correct. So Amit, basically we are checking that, we are cross-checking whether we are putting it in the right direction or not, like for the like, yeah. So we are just cross-checking and we are verifying, like to correctly orderize, right? Okay. Yeah.
Right. We are
And
just
also
cross-checking,
for additional
verifying.
risk, right?
The
Additional
only
risk
is
also.
that, sir, what I understood is that the only is that we categorize, we don't have to categorize, it is already categorized that it is important, potential, or missing.
Correct.
Means in order to restrict any other risk information into other orderized forms, so we are just using and checking out whether we are placing it in the correct box or not. Like, yeah, got it.
We are classifying it correctly or not.
Yes.
So even if the RMP is present, we will check for additional risk, right?
Right. If we will check actually what happens, the RMP version 1 covers the concepts. So for innovator molecules, RMP version 1 and PBRER 1 match exactly the same. But it happens, like suppose if SMPC is having additional risk, then we do need to check and get it confirmed from physician whether we need to include this risk or we can skip and keep it consistent with the RMP.
And also
these are all the questions to confuse you. Okay.
Yes. Also, Amit, we have to discuss with on kickoff meeting as well, right, with the physician and get it confirmed after that.
Yeah, that is the first part
no?
you have to do. Before kickoff, you have to do all this homework and then get it confirmed from physician. Until you have the endorsement from a physician, you can't go ahead with any of the risk. You can't take the responsibility for risk.
Okay, yeah.
Any other question?
So will they be asking about the causality? How do you do causality and all that in the interview, or just how do you pick the?
Yeah, they will ask you about how do you pick what is the parameters that you look to classify the risk. They won't ask as to how you look for causality because you can't find the causality in the RSI. There are certain parameters based on which you identify that, okay, this is the identified risk, this is the potential risk.
So this question
So
would
that's
not
why
be there.
no, for causality, they won't ask you a question. If they ask also, then you can say whatever parameters I will tell you in the next now whatever we will discuss as to how to pick the risk from the RSI. I'll tell you certain parameters. So those parameters you can explain to them, they will understand. They won't ask you as to how you establish causality for the risk from the RSI. That is not a question because nobody can do that. We just follow certain parameters to categorize the risk into those classifications from the RSI directly. If we get from all these sources, then it is fine. Otherwise, we have certain parameters based on which we classify those. And always you have to say a point in the meeting or means in an interview that you classify all those things, everything, and the endorsement is done from the physician. Final call. Simple.
Okay.
Yeah, Amit, I'm having one question. Amit, what is the difference between risk and this means risk and signal? Means both are same, or like what are the major difference between those two? Yeah.
No, if there are two, always remember that if there are two terms used, then definitely there is a difference between them. Okay. So just to tell you one thing, I'll say that just remember one thing, that every risk was sometimes a signal, but it is not necessary that every signal is a risk. Did you get my point? Matlab, har risk, whatever risk is there, it was a signal at some point of time. And then when the signal was evaluated and confirmed, it changed into risk. But it is not necessary that every signal will become a risk. Did you get the point? So now let me explain further. But did you get my point, what I told you?
Yes.
That every risk was
Yes,
a
sir.
signal at certain point of time, but every signal is not a risk. Okay. So now
how
what happens?
it will become a risk?
Yeah. So that's what I'm telling you next. So suppose you are doing ICSR cases or you are doing literature monitoring. In that you find, suppose adalimumab is known to cause serious infection. Okay. But now you know that they are causing serious infection, but at certain point of time, serious infection was reported as an event with the adalimumab, right? Imagine that situation some 20 years ago. Okay. What happened? Serious infection was reported as ICSR or from the literature source, and you find that serious infection was reported for the first time. You came across some 10, 20 cases, 5 cases, 1 cases, whatever is there. So at that time, that drug event pair become a signal for you. Means it is a new information for you, right? You're getting my point?
Yes.
So adalimumab and serious infection was a new drug event pair. It is a new signal for you. Now what will happen? You will start analyzing that signal. You will have to evaluate that signal. There are different steps that we discussed. First, adalimumab and the serious infection drug event pair is detected as a signal. Now we will validate. So we will do some database searches and we will see how many cases are reported and based on all those category, we will categorize this category A, B, C. Then we will validate and then we will go for complete evaluation of that signal, that adalimumab with serious infection. This we will evaluate. So what we will do in evaluation, we will look at different sources which we discussed in the starting of the call. We will look for preclinical studies, we will look for clinical studies, we will look for post-marketing cases, we will look for literature cases, we will look for class effects, we will look for similar labels, we will look for regulatory authority databases. Based on all this analysis, if we find a causality association between the drug or a susceptible causality association, means there is sufficient evidence to establish a causal association, or there is only scientific evidence, means you don't have enough cases in your databases, but there is literature available which says that it happens. So this is susceptible causality. So based on this, you will confirm that yes, my signal is evaluated and it is now accepted or confirmed. So once you confirm the signal, then it will become either a risk or a monitoring issue. So if it is becoming a risk, then it will has to be categorized into either important potential or important identified, or potential and identified risk. This is the whole life cycle of identifying a signal to turning into a risk. Did you get what I told you?
Yes, yes. Also, Amit, whether that non-serious events can also be a signal, right? And
Different,
it cannot be
definitely.
considered as a risk. So that's why you told here, like, risk can be a signal, but not all the signal can be a risk, right? So
No, I told this because every signal is not confirmed. They are refuted also. So they will not go into later stages. It will not be evaluated, or after evaluation also they are refuted. So they will not change into risk or monitoring issue. Okay. This is not due to serious, non-serious. This is because signal is refuted. So signal is refuted, so no point of changing into risk. But if it is confirmed, then it has either to be a risk or to be a safety monitoring issue. Monitoring issue is not a risk, but it is a flag for you that you need to monitor that. So keep it a different part. We will discuss it somewhere when we are discussing signals. So risk, just focus on risk. If signal is confirmed and it is changing into risk, then we have to see as to which risk it is converted, and we will update our RSI accordingly. So did you understand the journey of signal becoming a risk?
Yes.
Yes,
yes,
sir.
yes, Amit.
Okay. And non-serious information, that will also can become a risk for you, but it will not be important. Okay. And it might be just an ADR to be included into your RSI. It might not
Yes,
be a risk which has to go into the PBRER, but it will be included into your RSI if some some non-serious issue is there, event is there. Okay.
yes, okay.
So now you understand the journey of signal becoming a risk and what is the difference between a signal and a risk, right?
Yes, Amit.
Any other question?
No.
No questions, right? But still we have to cover the picking of risk from the RSI, and the time is over, I can see. And it is
Amit, I
also a
have one
vast topic.
question. Yeah,
Mhm.
yeah. So, what is CMDH basically? I just wanted to know what is the role of CMDH?
It is basically you can say use a committee of medicine for human use, something like that, decentralization. So that is basically a committee like PRAC is there now, Pharmacovigilance Risk Assessment Committee, who assess the risk or who assess the PBRs that are being submitted to European region to see if there is any new risk emerging. The same way CMDH and CHMP both are other committees also who look for the monitoring of this risk, and CMDH also work for veterinary, for human, everything. So they are broader perspective, but from human use, we basically consider PRAC, pharmacovigilance. So all these committees are basically involved into the monitoring of the molecule emerging safety profile in their region, in European region, basically, you can say. These are for European region. Okay.
So that means this is a collaborative committee which organized for basically to maintain this risk evaluation, right?
Right. To monitor this risk, whatever risk or safety
Okay.
profile is emerging for
Yeah.
any of the molecules registered in this region.
Okay, got it.
Means you can say these are the subsidiaries of EMA.
Yeah, right.
Okay. Any other question? People, any other question? Because I think we won't be able to take that RSI picking, picking of risk from RSI, because it will take another 1 hour or something.
Amit,
So we will discuss it in next now.
last question from my side. Actually, you told me in the starting of the session, like this new update about in October. So what was that? I just missed that thing. Can you just speak? Can you just tell us
October update was that na ki
what was the update?
last year EMA mandated that every MAH who is having their molecules registered in EMA region, they have to provide either a RMP to be uploaded on website for public viewing, or at least a RMP summary to be updated on the EMA website, that EPAR website from where you are looking those RMPs. Before October 2023, it was not available there.
Okay, thank you. Thank you.
So now it's easy for you guys to download those RMPs or RMP summary and check the risk.
Yeah, hi Amit.
Yeah.
So who is writing this RMP document? So it is a part of PBRER or separate committees writing on behalf
No,
of
we are
PBRER?
we only write this RMPs, the PV people only. So these
Okay.
are, see, in PV, there are different streams, you can say. Like you start from call centers are there, or data intake are there. Then ICSR is there. Then after ICSR, signal detection is there. Then this aggregate report writing team is there. Then RMP writing team is there. So eventually, as you move ahead in your career, it is better that you try each and everything. Like if you speak about me, then I have done all these things, and currently I'm mostly into the RMPs only. So this is how you will eventually progress into your career from call center, ICSR. Somebody might not be in the call center, they will start directly from ICSR. So ICSR, then aggregate reports, then signal management, then RMPs. Then there are other streams also. So you will eventually, these are all part of PV only.
Okay, yeah. Thank you.
Any other
Sir, one
question?
last question. These RMP and all those things will be provided to us, or we have to search it? Means like before writing a report, this all documents like RMP or something is been provided to. So we have to go
No,
and search.
like suppose if you are working in the aggregate team of a company, then they will give you access to RMP data, means RMP repository also, from where you can search whether the molecule for which you are writing the PBRER RMP is available or not.
Okay.
And if it is available, you can download that RMP from there and you can easily use it as a source. Any other question?
So if it is a initial version of PBRER, then how come RMP is there for that?
Yeah, so always
analysis?
remember that when you are going to apply for an MAH for post marketing, at that time you have to submit your RMP along in the NDA application. So your RMP version 1 is first prepared before writing any PBRER. So when you will when you will submit the RMP, at that time only you will get the approval, and if you have approval, then you will start writing the PBRER. So you have RMP version 1 handy at the time of writing first PBRER.
Okay.
Okay.
Means by default we will be having one RMPs with us, right?
Yes, if it
PBRER
is an
was
innovator
approved.
molecule.
Okay.
If it is an
Okay.
innovator molecule.
And what about clinical trial molecules in that?
Clinical trial molecules, you don't have to write PBRER, that we write
Yep,
DSUR.
DSUR. Yeah, so that was the other step, right? Okay.
Right. So that there is not a concept of risk and all. There is something, means risk is there, but that is not that elaborative as you write in PBRER. That is more into the clinical trial phases and all those things.
Okay. Yeah.
But remember, we are doing this training and you will be representing yourself in any interview as to you write only PBRER, because we are exhaustively discussing PBRER, and PBRER are the main document. If you are doing PBRER, then you can do any of the aggregate reports. So always concentrate and tell everybody that you are writing PBRERs only. If they ask for DSUR, then they say, don't know, I have not written any DSUR. Simply. Because without doing aggregate reports, you can means you can at least get a hands-on on one of the reports. So I have chosen PBRER because that is the most widely accepted report, and if you have experience in that, then people will take you easily, and they know that if you have written PBRER, then you can write any of the aggregate reports.
Sure.
Got it.
Sir, one general advice means like all this recording can we means like we can incorporate all the recordings on Google Pathway and give it access to only those people who have been added into that group, so that we can go like yesterday we while revision we faced an issue, but it was corrected.
So you can guide me on this as to how we can collaborate and we can move it from Zoom site to our own Google Drive or anywhere where we can give access to the people who are in the our group so that they can use it, because I have not done it, so I'm not sure as to how to do it. So if you can guide me, we can definitely do that.
Yeah, we can put all our files and all
Yes,
shared
Mhm.
files there.
Amit,
Okay, we can do
today
that.
also I just called you about regarding actually I'm not having the WhatsApp right now, so yeah,
Uh-huh.
you will be sharing me another WhatsApp number. So if it will be very easy for us to just refer and go that way. Yeah.
Okay.
I'll,
Okay, so we
I'll
can do it, but
I'll check Amit and I'll let you know because I think we need to give the access to the mail IDs
okay.
and
Sure,
access
Sumit.
this links.
Sure, you can check and let me know. We can make it at a collaborative platform, because any which ways, if we are losing the Zoom access, then we can lose the recording. So if we can have this on our pathway from where we can easily access, then it would be great.
Okay.
Any other question, people? If no question, then how is the training ongoing? How are the sessions? Are you people able to get, because you're not working on these documents? So are you able to get the
Actually,
concepts?
Amit, I'm talking about me. I'm not getting the time to practice actually. So whatever we are discussing, like that only part only I can understand, but actual writing of the report, I'm not getting time.
So this is the prerequisite, because you will have to take some time out, because see, we are busy, but if we are trying to move somewhere or we are trying to move out of the box, then we will have to dedicate some time. So at least half an hour also, if you can dedicate in the weekdays, then you will definitely, mean, suppose till now we did 4 sections of the PBER. So at least half an hour you can spend on each one section, whatever you have written, suppose 4 lines, 5 lines, 6 lines, it's fine, leave it. Then come another day, write another 4 5 lines of that section and complete it. Then maybe one section you can complete in 2 days. So that ways you need to take some time out of it, because I can't help in getting time for you, right? You only have to work on that time part. So you can, means, get up early in the morning if you're not getting time during the days, or you can sleep half an hour late in the evening if you're not getting time during the days. So that you have to work out. See, I'll tell you my story. When I was in ICSR, I was looking to move into the aggregate. So you can understand the same thing I was facing, but I didn't have the mentor at that time. So what did I do? I read through GVP modules. I got some PBER references. I catch hold of some person who was in my project. Means, when I moved to compliance at that time, I had some people who were from aggregate report. So I learned from them little concept. Then howsoever I managed to move with the IJP into the same organization in TCS, also into the aggregate. Then when I moved into the aggregate, then I formed a group of people where we learned the niche concepts and everything. Then we moved out of TCS and eventually. So you have to take initiative for your development. So you'll have to take some time out, because if it is a burning desire inside you to move into the aggregate, then you will definitely get time out of for yourself.
Hello,
Okay,
Amit.
thank you for the motivation.
Yeah, no problem, but you have to take time. So please practice. Otherwise, just discussing the concept in this classes, you will forget for sure. So this concept which we studied today, definitely if you'll not revise it during the weekdays, then you in the next class you will again be jumbled as to what happened.
Amit, can you just give us some reference where we can find that PBRER report which is available in the open source in our internet? Because I'm
For
unable to
PBER,
find. Yeah.
Yeah, for PBER, there is no open source, or like EPAR, you are there for RMP. So for PBER, there is no such website. You just have to randomly search into the Google if you can get the PBERs. And the PBERs which will be available, they will be very short form PBERs only. So means you will not get that insight. So you have to focus on the download all those PBERs just for your insight, look into those. But whatever I am training you in this, keep practicing those sections. Therefrom only you will eventually evolve as a good writer. So keep practicing, download some papers, but I'm sure that you won't get that materialistic paper. You can find at least one or two. Search for Apixab and PBER, search for what you can say, some Roche papers are available. I think you can search for that. So randomly you have to search on Google to get those papers, but there won't be much information in those papers.
Hello, Amit sir.
Yeah, tell me.
Yes, you said that we have to read that GVP models, right? So I tried reading that, but by reading that, all there are many questions and we don't get clear by reading that. Are any videos which are related to GVP models which you can share, which we can get more concept clear?
No, from the modules, it is your question is correct that you don't get clarity by reading the GVP
Yes.
module. That's why I told you that use it as a reference and pay attention into the training. So you will understand what is now. Suppose 4 sections you have done. So when you will go to GVP module section, now you will understand it better. After that, 5th section, 6th, 7th, 7th, whatever sections post 4 are there, if you will read, you will get little concept. But for 4 sections now, if you go, you will understand, okay, these things we studied. So this is how it goes. So further, when I'll teach all the sections, then you will go and read those sections into the GVP, then you will get the concept. So these are the guidance documents. These are just for guidance. These are not the Bible, you can say, that step by step you have to follow like that. And they won't give you much elaborative explanation also. That is a part of MAH. So it comes with the experience. So collaborate means combine both the GVP module and the training and try to understand the concepts.
Okay.
Hi,
But there is no
Amrita.
such video. No, there is no such video which could explain the GVP
Yes.
modules.
I was searching for that only yesterday. I searched, but I didn't get any video which we
No, you won't get any video. So that's what I told you, that just go through GVP module and pay attention on the training. So this training will become a video, whatever is a necessity for video now. So this training is becoming a video for you, you can say.
Yes.
Yes, next question. Who was asking something?
Yeah, Amit, I was asking that let's say if we clear the interviews, okay, and we go into the company, so what will be their expectation? Like, will it be overwhelming? Like, right from the start, they will ask us to write the report and all that.
So see, if you means go and join a company and go there, then what they will do, any company will be there who will give you a training first. Okay. So training will be lesser than this. Whatever we are studying here, they will just give you an overview. Means they will give you a template. They will show you a template and tell you that because they know that you have an experience onto the aggregate writer. So the training is conducted to complete their curriculum part, which is a part of the training and management session for as per the audit system. Okay. So they will train you. They will keep you for 1.5 months into the training. So in that training, you will have access to their keywords and everything. And now you are trained here. If you have trained here and you got the concepts, then only you were able to clear the interview and test and went to that organization. So you have a fair understanding of the PBR sections and everything. Now you have the PBRs. When you join the organization, you have the PBRs in front of you. So you will start reading at the time of training. What you have to do, you start reading the PBRs, look at the source documents and everything. And when they will give you the document, at that time also, don't worry. The document, what happened, they will give you the second PBR, third PBR, fourth PBR. This will not be the first paper when you are writing first time. So this will be the fourth, fifth paper. So you have a previous paper already there with. So you just have to look into the data, the process and everything. So for the first 2-3 papers when you are writing, you will have just previous papers based on which you will be writing. So you will be acquainted with the paper while writing the first 3 or 5 papers, and then you will pick up from there. So don't worry that if I don't have experience. But if you have studied properly here, you have practiced properly here, you have cleared the interview, then you are at par to work in that aggregate report writing team. Don't worry about that.
I mean, will they ever suspect like we don't have the
No.
experience or
No, it depends on you, means how you are presenting yourself. Okay. When you go there and you will say like, "Arey, ye to mere ko nahi aata, ye nahi aata," then they'll suspect na. But if you work hard on your own, you means when you are working there also, then also you will be having access to me. Okay. If you have any doubt, you can ask directly to me. I'll tell you. No, And when you have worked for 2 to 3 PBRs, I can assure you that if you have worked on 2 to 3 PBRs only, and you will be much more confident, nobody will suspect, nobody will find that you have not worked earlier. Only 2 to 3 PBRs are the first means you have to handle properly. And PBRs doesn't require much, you can say much skill. More of it is the process that you have to follow. The process is stringent. The timelines that we discussed, the timelines that you have to follow. Aggregate report management is more critical part than writing. Writing is fine. You will 2 to 3 PBRs you will write, and you will know the process as to how we need to write. But the timelines management, the QAMs, the stakeholder management, these are the important things.
Amit sir, we will also write 2 to 3 papers right after we finish the training.
Yes, we will write. I am arranging the line listing. Some I have got, some I am arranging more. So we'll do that.
Okay.
Don't worry.
Also,
And
can
only
we
you will
know?
have the.
Also, can we know what actually in day to day what aggregate reporter do? Actual work.
That's the thing. Means the timeline preparation I told you and all those things. Then writing the sections, how to means separate the cases in line listings and all. These are all the things that aggregate report writer is doing. So whatever is
So-
being done in the training, it is the part of aggregate reports day-to-day work.
So we will start from 90 days before the DLP. So before that, what aggregate report will be like right from
Like suppose if you are assigned with the first, means you went into an organization, you got your first paper to write. So you'll start the work on 90 days as per the timelines. And then whatever is listed there, like before 90 days, you will get a stakeholder confirmation. Then you will send the data request, like line listing request, regulatory authority request, regulatory affairs team request, sales data request. These all requests you will be sending in between. In between, you will be preparing your KM document in which you will prepare, you will identify the risk. You will identify means, or you can develop the search strategy for those risks. As we progress into the report training, you will come to know that in QM, you will identify risk, you will make a strategy for that risk, the search strategy for that risk, how to present those cases. Earlier PBR, you will see these are presented in this manner, the same manner we have to present here also. You will take the physician confirmation, then you will get the draft line listing. Once you get the draft line listing before the DLP, you will start writing the PBR. Then you will get final line listing, DLP +7 or DLP +10. Then you will draft the final report with that. Then you will initiate the physician review for that. Once physician reviews that, you will address the comments. You will then initiate the quality review. Quality review is done, then you will initiate the approval and everything. And the same way, the submission will be, you will file for submission. So these are all the things that you will be doing in the 6 months time. On one paper, you get 6 month time. DLP minus 90 days plus DLP plus 85 days, approximately 6 months, 5.5 months time, you can say.
And by
apart from this work, apart from this work, do they ask for other work to do? Like, no, if we are
See,
not
as the
writing any.
right, suppose like you are in ICSR, so some people would be doing compliance activity, some people will be doing reconciliation activity, some people would be doing allocation and all those things. So the same manner the aggregate also functions. If you have some space or you want to work some extra mile, then you can ask for some additional activities they will give you. Do the literature screening, do the compliance activity, reconciliation activity, or maintain the master tracker for aggregate in which we do the assignment and everything. So these are the different work they can ask you to do depending on your interest.
No, in ICSR, we are forced to do some other work with ICSR actually.
So here what will happen, that if you ask for certain work, then they will give, otherwise aggregate itself is having, like at a time you won't have one report only, you might have 3 reports of different DLPs, means 2 reports of DLP within a month. So you will be engaged, don't worry about that.
Sir, I had a general question. Like we people are experienced in ICSR, then also most of the time they don't take the people from ICSR means like we do have an experience. तो ऐसा क्यों होता मतलब is it like very much complicated so that ICSR people cannot work, or like what was the reason behind it? Means some of the time I see that in the note that ICSR experience will not be entertained. People don't like apply if you have an ICSR experience. So sometimes it is demotivating because it is the part of PV and we do have such a experience in, then also they write like this. So is it very much like complicated so that they want the reason I don't understand why it is like this.
Okay. So tell me one thing, like suppose you are currently in ICSR, right?
Correct,
And
sir.
you are processing the cases, quality reviewing the cases, but you don't have any experience into the submissions. Okay. So will any company take you into the submissions team directly?
No.
Why no? Because there is a risk that submission teams had to submit the cases directly to regulatory authority. And if there is any issue, then that will fire back to MAH, right? There is a risk involved. So that's why they don't prefer only ICSR people. If you don't have submission experience, they won't take. Second point, till now, whatever training you have attended, before attending this training, you tell me, if anybody could have asked you this concept, would you have been able to answer them?
No.
These are all part of PV only, right? Just
Correct.
for an example, till now you know that ADR, but today you learn the concept of risk. Okay.
Risk.
So, but earlier, if people ask you what is the difference between ADR and risk, would you have been able to tell them? No, right?
No.
Because you don't have experience into that. You don't have experience. Now you are attending the training, so you are understanding the concept. So now you can explain them. So why we ask for experience into the aggregate? Because aggregate is a compile. You are working in ICSR, you are working on single cases. You would be having some target 10-15 cases in a day. You are currently, what is your thought process? You are into a single patient case. You are doing that. Okay, what is the suspect? Was the event? Or how do we do the coding? What is the causality? How to find? Or causality not? What is the listlessness, labelness if it is available? Some are having automatic. So these are all your thought process that. But if you are working in aggregate, then your thought process is different. You are working from minus 90 days. You are working from minus 90. You are working on strategy document. You are working on how to strategize your report. So your thought process changes. Okay. That thought process won't be there if you are working in ICSR. So now when I am working for an aggregate report, so when I'm looking for a candidate into my team, I would prefer a candidate who is having an experience into the, be it 6 months, be it 1 year, be it 2 year. As per my budget, I will look into for the experienced people. I won't look for the ICSR people directly, right? कि मैं ICSR को लेके I'll train. I don't have time to train them. So I would look the person who has at least worked for 1 year, 6 months, or 6 months. So I am just telling 1 year, or at least 1 year or 2 year. So that is the thought process behind. That's why don't get more demotivated that they don't entertain ICSR people. It is nowhere written that they don't want ICSR people. They are saying that we need experienced people into aggregate. So that experience you have to develop. It is not necessary that you are working into the aggregate reports. You need to have that zeal to develop that. And if you are satisfying them in interview and everything, then they are okay. They are not looking as to whether you have done aggregate or not. Did you get my concept? Nobody will entertain a person from ICSR into aggregate. No company does that. You won't come across any company who takes, if you say that you have ICSR experience, they will take you into your aggregate. If you say at least 1 year into aggregate, they will take you. They are ready to take you.
And it depends on interview actually, and how you showcase yourself in the resume, and then how is your interview.
In the interview,
Totally
differently.
depends.
Interview and the written test they take. For lesser experienced people, 1 year, 2 year, they take written test. In that written test won't be that complicated, but they will take written test. Based on these two parameters, they will decide whether they have to take it or not.
Hello.
Yeah.
Hello.
Yes, tell me.
Hi Amit. अभी सिचुएशन कैसा होता है कि डेजिग्नेशन भी मैटर करता है, तो वो कैसे
right.
हैंडल करेंगे आफ्टर इंटरव्यू के टाइम?
देखो आप ऐसा रहता है ना, व्हाट हैपेंस कि द lesser experience you have, that doesn't matter, or the more experience you have. What matters in any organization is the budget. Suppose if you are having an experience of 11 to 12 years and you are working as a manager in ICSR processing, but your CTC is less than 10 lakhs, and you present yourself for aggregate interview as that you have worked in ICSR for 8 years, and from the past 2 years you are working in aggregate. So since you are working from past 2 years, so they will look to accommodate you at the lower level of aggregate. Okay, they won't give you manager at aggregate, but they can give you a grade below that. So you will have to compromise with that grade, and your budget will fit into their pocket. Then they will interview you. So that is a simple funda that works. But you will have to compromise somewhere. Like you can't say that I am a manager here, so I'll go as a manager in aggregate. So then they will expect at least 7 to 8 years experience into aggregate. So that is the way how you need to compromise initially.
ओके, अगर सपोज मेरा एक्सपीरियंस अभी 8 इयर्स का है और मैं ऐसा इंटरव्यू में ऐसा बोला कि मैं लास्ट 2 इयर्स से एग्रीगेट कर रहा हूँ, तो वो मीन्स अगर मैंने परसेंटेज ऑफ हाइक मैंने अगर इतना पूछा, तो वो उसमें HR मीन्स जितना मैं एक्सपेक्टेड हूँ उतना नहीं दे सकते ना?
देंगे. देखो, if you are lesser, means aggregates budget is higher. Okay, if just for an example, if a person is working into aggregate for 8 years, so at least he would be somewhere around 15 lakhs at least. So when you are into ICSR for 8 years, then you would be, as for my understanding, you would be lesser than 10 lakhs. So you already have a margin of 5 lakhs. Okay, so you will ask for 30%, 40%. So 30-40% everybody can give you easily. There is no bargaining in that.
Okay.
So
Actually,
experience
PV
doesn't
में
matter, right?
Yeah.
So experience doesn't matter in respect to hikes. Like if they have budget, they will give you.
Actually, currently in ICSR now we are facing such issues. Means they are